Good morning, and thank you for joining us for a live talk with STEM Ambassador Mark, who's an Associate Professor in Cellular Neuroscience at the University of Reading. It's great to see so many of you joining us today. And to celebrate British Science Week, Mark will talk about a day in the life of paracetamol, detailing its life cycle and how it goes from a pill in your hand to a remedy for your discomfort. Please feel free to put any questions in the chat and Mark will aim to answer as many as he can at the end of his talk. So let's get started. Good morning, Mark.
Good morning, Lisa, and thank you very much for the introduction. So welcome everyone, and I hope everyone's having a good British Science Week. So yeah, I'm going to take you on a little bit of a journey about a day in the life of paracetamol, and maybe, as Lisa said, take you on a journey from that pill to giving you that kind of relief that you're wanting from that particular medicine. But before we kind of dive into paracetamol, just a little bit about me, how I became a kind of scientist and what's kind of driven me through my kind of career. So I originally kind of studied the sciences at school, kind of chemistry, biology and physics. I think probably biology kind of stood out as the subject I most preferred and really kind of enjoyed learning about it and finding out more about the kind of world we live in. That kind of drove me on to think about structure and function, really about the human body. So I went off to kind of University of Dundee in Scotland and did a BSc in anatomy and physiology. Having been to kind of university for 4 years, as you do in Scotland, that kind of really opened up my eyes to thinking about a little bit more about the brain and how the brain operates, and really how we don't really know that much about the brain. So I then went off to the University of Leeds, so migrated south to Leeds, and that's where I did a PhD in neuroscience, starting to understand some of the neuronal pathways, some of the cell connections within our brains and how they maybe dysfunction in certain disease conditions. And then that led me on to taking up a position here at the University of Reading, where I'm a lecturer. So I do a little bit of teaching, but also do a little bit of research as well. So I'm a neuroscientist by training. But today I'm going to tell you a little bit about kind of pharmacology and how that impacts our everyday life. So if we think about pharmacology as a subject, what is pharmacology? So pharmacology is the study of drugs and how they operate in various systems. So it might be the human body, but it might be maybe pets or animals. You might be thinking about kind of veterinary medicine, but also we need to think about how animals interact with drugs as well. And under the kind of umbrella term of pharmacology, there's pharmacodynamics and there's pharmacokinetics. So pharmacodynamics is really what that medicine or what that drug does to your body. So thinking about the effects that it has and whether that's a therapeutic effect or indeed whether that's something that's an adverse effect in terms of maybe drugs of misuse. On the other side, we've got pharmacokinetics. And that's the kind of study of the processes that what your actual body does to that drug. And that's what we're going to focus on for this morning's talk. So thinking about once you actually take a medicine, what does your body do to that? And how does that maybe work well in terms of actually making that drug a better medicine? Or indeed, maybe it actually impacts on how often you might need to take a medicine or how much of a medicine you might take to get that particular therapeutic effect. So the drug we've chosen for this morning is paracetamol. So if I was with you in the school or college and asked how many people had taken paracetamol, there might be a lot of hands that have been put up. But if you haven't taken paracetamol, I'm sure you know someone who has. And so this slide here just shows the chemical structure of paracetamol. And if you look at paracetamol as a pure product, this is what it looks like. But this isn't what we take. This isn't what we actually administer to ourselves. We actually take little things that look like these tablets, capsules, whatever you get from your pharmacy. So there is a process by where we make the molecule and the powder into a medicine. So the medicine contains other ingredients alongside the active ingredient, the paracetamol, to make it, on this box it says, easy to swallow. So again, you're thinking about making that medicine into something that someone can take by swallowing in their mouth. And if we think about what does paracetamol do? And so I mentioned that some of you are thinking about maybe have taken paracetamol. So paracetamol is the leading analgesic drug. So it gives us pain relief. So it tries to, in essence, reduce the pain signaling within our bodies. And this graph here just shows that far and away it's the most dispensed items. So the number of items that pharmacists give out under this class of drugs. And again, paracetamol is used to monitor or manage pain, and in particular fever. So whether you've got a high temperature, you try to, in essence, take paracetamol to reduce that pain and fever associated with it. So that's why it's important that we understand what paracetamol does, but also how our bodies respond to paracetamol, so that we know that all these people that are taking paracetamol are taking it correctly, and it's not having any adverse side effects. So this comes on to pharmacokinetics. So the four kind of things that we want to take away from today's session are this mnemonic, ADME, A, D, M, and E. So what do they stand for? So we've got absorption, distribution, metabolism, and elimination. Simply put, absorption is about getting into the body. So how do we get that paracetamol into the body? Distribution, once it's in, where does it go? How does it move around your body? Metabolism, what are the processes that start to break down that paracetamol molecule into something that's more amenable to the final stage, E, elimination, and getting rid of that medicine. We don't want the medicine to hang around for a prolonged period of time, because that may actually have adverse effects for individuals that are taking that particular medicine. So we want it to get in, do its job, and then be eliminated from our bodies. And one way that we can actually monitor pharmacokinetics, and one way we do in the laboratory and in clinical trials, is to monitor some of these parameters. So what here is a kind of blank graph, and we'll start to fill it in as we go through the talk this morning, is we have plasma concentration. So this is the concentration of the drug that is in the bloodstream, circulating in your blood, and time here. So British Science Week theme this year is time. So again, we're looking at the time factor here. So we want to monitor how the concentration of the drug changes as a matter of time, and understanding how those processes, ADME, absorption, distribution, metabolism, and elimination, feed in to actually this graph as we go through this morning. So let's deal with absorption. So absorption, as we said, really relates to how much of that medicine is going to get into the bloodstream. And this ultimately depends on the route that the medicine is administered. So you can start to think of the different ways in which medicines are administered. We looked at pills in the first couple of slides. So they're taken orally, so you swallow them. But there's other drugs that sometimes are injected. So again, how you take a medicine actually impacts the level at which you would see in the bloodstream. And again, the parameter that we measure in the laboratory is bioavailability. And that's how much of a drug that's administered actually reaches the bloodstream. And again, one of the things that you might not be aware of is that most of that medicine that you take your paracetamol, not all of that paracetamol actually then goes in and reaches the bloodstream. Your body's already starting to break it down, and therefore that has implications in terms of how much paracetamol you need to take in order to get something or a dose in the blood that is actually of benefit to you. So if you think about just doing a quick calculation, and most of the paracetamol tablets that we take are approximately 500 milligrams. And if we say that the oral bioavailability, and that word oral will become apparent in the next slide, is 88%, or 0.88 as a fraction. And how much of that makes it to the bloodstream? So therefore, only 88% of that 500 milligrams of paracetamol that you've taken actually makes it to the bloodstream. So therefore, what ends up in the blood is this 440 milligrams. So we're losing 60 milligrams of drug. So where does that go? So that's our body starting to kind of break down the paracetamol. That means that it doesn't get a chance to pass into the bloodstream. So even though you're taking 500 milligrams, what actually makes it into the blood is only 440. So we need to be aware of this bioavailability, and that can range from anywhere between 5% and 100% for different drugs. And that tells you and has implications in terms of the dose, the amount that you need to give someone, so that you get a level that's actually going to bring some therapeutic benefits. So if we look at our graph, so now we're starting to plot absorption. So we've taken our drug here, and there's going to be a bit of a lag time as it passes through our stomach, through the gastrointestinal tract, and then we start to detect it in the blood. So this is through the oral route. Also highlighted here is if you inject it, it goes straight into the blood. So there's no absorption phase. So often if you think about where and when you might be injecting drugs, that tends to be kind of in hospital, when you need that drug to be into the blood very quickly. So therefore, oral route is much slower in terms of that absorption until you get levels within the plasma that are effective. Distribution. So we've done the A. We're thinking about now how far that medicine can actually reach within the body itself. Does it just stay in the bloodstream, or does it penetrate out into different body compartments? And again, different medicines have different abilities to penetrate throughout the human body. And this has implications on the dose and the levels of drugs that reach the bloodstream. So if you imagine our body being this kind of bathtub, and we're putting in 2 tablets, so normally we take 2 tablets of paracetamol, 500 milligrams, and we're dropping it into our bathtub, and this is an example of a human body at 1 litre. So therefore, you'd expect the concentration to be 1,000 milligrams per litre. But our body isn't as simple as this bathtub. About 70% of our body weight is made up of water, but that water is distributed into different compartments. And different drugs can reach those compartments. And that might take some of the drug away from the bloodstream itself. So this highlights here the 2 extreme cases. Here on this side, we've got the drug concentrated in the bloodstream. So it's got a low level of distribution or volume of distribution. It's retained within the blood. And that's good for maybe some drugs that you want to act on the bloodstream. So such as warfarin, which is an anticoagulant, it stops the blood clotting, and therefore that needs to work in the blood. On the other side, we've got the other extreme, where that drug is able to penetrate out into some of the tissues in the body, such as fat, adipose tissue, or indeed bone. So again, there's a range of distribution that occur. And for paracetamol, it's 65 litres. What does that mean? That means it kind of actually disperses throughout most of the body. It's not restricted to the bloodstream. So what happens after distribution? So again, we've started the absorption. We're starting to distribute the drug, sorry. And so that might mean that actually we've hit a peak. So some of the drug is then actually moving out of the bloodstream and into some of those other organs and tissues. So therefore, we'll no longer be able to detect it within the plasma itself. Think about M, metabolism. Our bodies are great at metabolizing a whole range of different chemicals that we choose to ingest, whether that's medicines or whether that's drugs that we might be taking maybe socially, or indeed drugs of misuse. And our body has these unique family of enzymes that can actually tackle a range of different chemicals. So the family that we're interested in here is this cytochrome 450 superfamily. And indeed, paracetamol has a specific enzyme that's involved in breaking down paracetamol. So there's a whole host of these different enzymes that are uniquely able to tackle individual molecules. And so they're looking at breaking down the paracetamol molecule into something that's more manageable. And we think about more manageable, what are we trying to actually afford? So we want to make it ready for getting it out of the body. So again, we're looking to increase water solubility and also decrease biological activity as well. So that transformation from the parent molecule into a metabolite is getting it ready for your body then to excrete it from the actual system itself. So we say decreased biological activity here. It wouldn't be science without some metabolism actually increasing biological activity. So there are cases where actually we need to know what this process is because we see an increase in the biological activity. And that's of interest, particularly in pain medicine and morphine, as we were mentioning previously. So metabolism. So what's happening on our graph in metabolism? So we've kind of hit a peak, and now we're starting to, in essence, break down the product, so we're losing concentration. So again, metabolism really starts as soon as the drug actually hits the bloodstream, but this is where absorption is exceeding metabolism, and this is therefore when metabolism starts to reduce levels of the drug in the plasma itself and break down that parent paracetamol molecule. So we're on the way down, we're on the way out. How do we get rid of that drug that we're taking in terms of paracetamol? So again, there's a whole range of different routes that a medicine can take to exit the body. Urine is probably the main one, but the sweat, saliva, exhaled air. This is particularly important in terms of breathalyzers. So in terms of when the police breathalyze people, they're looking at the amount of alcohol that's contained within exhaled air. And we know and we can calculate how much that relates to the blood concentration of alcohol. And there's hair as well. So some of you might be keen CSIs, crime scene investigator or watchers. And again, you can look at hair samples of deceased to understand their drug history, to understand what drugs they've maybe taken through kind of hair samples. But the main one in terms of medicines that we take in everyday life is urine. And again, you can monitor excretion through the urinary pathway. So how does that work? So again, the main organ that plays a role here is the kidneys. So the kidneys are like a filtering system. So they aid filtration of different molecules, and that's based on the size. So again, one of the factors in making a medicine is the size of the molecule. Size matters when it's making a medicine. And again, some of the products that are passed through the kidney actually are then secreted into, in essence, the urinary tract for excretion, but some of them are actually taken back into the bloodstream. So there's a fine balance between your kidney recognizing what it wants to excrete, but also in some cases, the drugs mimicking our biological systems to almost trick the kidneys into taking it back in so that then it goes round the bloodstream again, and we might get a prolonged effect in terms of that therapeutic effect that we're looking for. So again, back to our graph, what does it look like? So again, we've started metabolism, and this is also part of elimination. And why is it a day in the life in terms of paracetamol? Well, this whole process for paracetamol, in terms of absorption to elimination, takes 24 hours. So again, the next time you're taking your dose of paracetamol, 24 hours later, there's going to be minimal amounts of that paracetamol still within your body. Interesting in terms of reading off these graphs, what do we look for as scientists? So if we start to think about what this graph looks like, again, we can start to then look at the timeline of different parameters. So if this dotted line here is our therapeutic concentration, this is when we're going to see benefit to the individual taking the paracetamol. So it needs to be above this level for it to be effective. So again, these blue lines here actually indicate the therapeutic duration, how long that medicine stays above this line and gives you that benefit for the paracetamol. We can measure the peak plasma concentration, so this is the maximum amount of drug that was in the bloodstream at a given time. And then we can also plot on this, what is a maximum safe concentration. So again, how much would actually then lead to maybe adverse side effects? And this is important to know, because if you take another dose around about here, then the chances are that then this dose will go up again, and it might then start to enter or exceed this maximum safe concentration. And so why is it important to understand pharmacokinetics? Well, one thing you might not be aware of is that there are people that actually die from overdosing paracetamol. It might not be a very big number. This is like data from 2021, but 227 people actually die from poisoning paracetamol. And that's because they've taken too much of that paracetamol and it's entered into that non-safe levels. So by understanding pharmacokinetics, we can better understand and better inform these individuals about how they should be taking the medicine to try and, in essence, reduce this number, ideally to zero, so nobody is actually being impacted. So hopefully that's given you a little bit of an insight into a day in the life of paracetamol and actually started to explore pharmacokinetics. So that's how your body tackles the drugs that you ingest. And each kind of medicine that you might be taking or you might see in the pharmacy actually has different kind of profiles in terms of the bioavailability we talked about, the distribution, how far and wide it goes in the body, what enzymes are involved in terms of metabolism, and how quickly it's excreted. And again, clinical trials are important in terms of that pharmacokinetic data. That's the first thing that they look for in terms of that data set. So again, the study of pharmacology and pharmacokinetics is important for all budding healthcare professionals, and hopefully this one or this talk here has inspired you to find out a little bit more about pharmacology and pharmacokinetics. So thank you for your time this morning. I hope you enjoyed the session and happy to kind of take any questions that there were this morning from the audience.
Thank you, Mark. That was really informative and interesting. And we've got some great questions coming in. So I'm going to hand you over to my colleague, Sophie, and she will put those to you.
Thank you, Lisa. And good morning, Mark.
Good morning, Sophie.
No problem. So our first question that I have got through is, does paracetamol help with pain or is it a psychological effect?
Yeah, so maybe the psychological effect that the questioner is referring to is the kind of placebo effect. But we know in terms of the mechanism of action that paracetamol does relieve pain. So again, we know through molecular targets and clinical trials that actually there is a biological response over and above that kind of psychological effect. Yeah, any drug has that kind of psychological effect or that placebo effect in terms of giving someone a pill. It could be a sugar pill that makes them feel better because they think they're actually making a difference in tackling that particular ailment that they have. So there is a biological response, but certainly there is a psychological response to any medicine, including paracetamol.
Okay, that's great. Very interesting to know that. Okay, so our next question is, are all brands of paracetamol the same that you buy from, say, the supermarket or the chemist?
Yes, so in terms of regulation, yes, every kind of brand is the same in terms of what's in it. So again, it has to have the active ingredient, the paracetamol, and again, it's a little bit of kind of marketing in terms of the brand, which one you prefer, but all of them contain the same amount of paracetamol. Some of them might have a different formulation, some of them might be made differently to make them easier to swallow, as the packet might say, but in terms of the actual medicine, the benefit you will receive, they're similar across the brand. So yeah, the cheaper ones are as effective as the more expensive brands.
Okay, that's great to know then. So the next question I have is, how does paracetamol reduce fever?
So in terms of reducing fever, so we know some of the enzymes that are involved in kind of temperature regulation within the body, and it acts as an enzyme inhibitor. So we know in terms of some of the kinetics and the enzyme pathways, what paracetamol does, and that what it targets is to reduce some of those, in essence, inflammatory pathways that lead to that build-up of heat in terms of how you feel fever. So that's one of, going back to the previous question, that's one of the biological targets. That's how it actually makes that difference in terms of a biological response.
Okay, I've got a question here. Can I study pharmacokinetics at university?
You sure can, yeah. So pharmacokinetics is under the kind of umbrella term of pharmacology. So again, you can think about going on to study pharmacology. So there's many degrees that you can do pharmacology, and that's kind of aiming towards something maybe more lab-based, but also healthcare professional. So if you want to be a doctor, a nurse, a healthcare professional, a pharmacist, they all study pharmacokinetics because it's really important that they understand the impact of the drugs that they are dispensing or prescribing has on that individual. And thinking about more complex scenarios, what are those drug-drug interactions? So we've just tackled one drug today, but think about maybe people taking multiple drugs and the impacts those drugs have on each other and have on the body's ability to actually regulate some of the pathways that we've talked about this morning.
Okay, I've got a question that's just come in. What is the safe amount to take?
For paracetamol?
For paracetamol, yeah.
Yeah, it's 500 milligrams, so it's a gram, and 4 times a day, and I think it's normally 4 to 6 hours that you meant to space it out. But always read the packet. So again, a lot of the pharmacokinetic science is actually translated into much more kind of lay language on the patient information leaflets. So those information leaflets that are stuffed in the boxes of medicines that we don't tend to read, open it up and read it. It will tell you much of what I've told you this morning in terms of paracetamol.
Okay, that's something that maybe everyone can do, is have a look at the information on in their packets of paracetamol. Right, yeah. The next question, I'm just having a look now. Oh, can you get paracetamol as a liquid or a spray?
So I don't think you get it in as a spray. I think you can get it as a liquid formulation. So again, if anyone's got any younger children or younger siblings, there is paracetamol in liquid form. Because again, thinking about pharmacokinetics, not everyone's able to kind of swallow pills. So liquid formulations are much easier for the younger generation, but also for the older generation. So maybe people have got elderly kind of grandparents. Some of them might struggle to swallow kind of pills, and so there's liquid formulations there. But I'm not sure about sprays, but again, it's thinking about different formulations that can open up avenues to people who can't currently take the medicines that are available. But it's an interesting idea to develop something along those lines.
Okay. I've got another question here. If someone weighs more than another person, do they need a larger dose?
So it depends on the route of administration. So in terms of a lot of drugs that when you go into hospital are given per kilogram of weight, so it's kind of a dosing per kilogram, so you weigh the individual and you give that amount. In terms of paracetamol, the range is not significant to make a difference in terms of the therapeutic effect. So whether you're kind of a difference in weight, it won't make an impact in terms of paracetamol. There are drugs that are taken that you need to account for that difference in weight. So there are medicines that does take into consideration, but for paracetamol, it's not pertinent to think about, because if you think about then having to weigh every individual, then having to then actually administer, so giving them just that one standard dose means it's much better compliance in terms of people taking the right dose and not having those kind of issues around kind of overdosing.
Okay, so it's for our safety then. That's why they're set at those standard doses. Okay, so the next question I've got is, can you give paracetamol to animals?
So you can. I'm not a vet, but yeah, you can certainly give paracetamol to animals. It would probably be maybe in a liquid form or in an injectable form. But then you need to start to think about pharmacokinetics of that individual animal. So I said like, our total body water is about 70% of our body weight. What animal are you wanting to give it to might depend on some of those processes we've talked about, particularly about that distribution, how, in essence, that drug is going to be distributed. And also thinking about elimination, how big the kidneys are and how efficient the kidneys are at filtering processes. So you can certainly give it to an animal, but you'd want to maybe consult your vet before thinking about kind of administering it yourself.
Okay. So I think we've got time for maybe one or two more. So if anyone's got any questions they want to get in, now's the time to pop them in the chat. The next question is, as a lecturer, what do you like most about teaching?
That's putting me on the spot. So I think in terms of what I enjoy most about teaching is that kind of journey that students go on. So some of the students that are kind of less confident in their own kind of academic ability in particular topics. So say I teach pharmacokinetics at university. Some of them kind of struggle with some of the basic concepts, but throughout the year, throughout the lectures and workshops, then they get a chance to actually present some of their knowledge and some of their understanding through assessments. And it's seeing those individuals that have kind of struggled in the class actually then kind of actually learn and actually understand it. So again, there's a lot at university about kind of retention of detail and memory. It's more understanding it and being able to kind of apply it. And that is stereotypically, it's kind of that light bulb moment when a student actually understands the concept and can apply it to any different setting. So we've talked about paracetamol here, but if you think about a pharmacist or a healthcare professional, they need to know for every single drug that they might be dispensing these kind of different parameters. So I think it's seeing those kind of students actually grow in confidence as they build up their knowledge base to actually then make a difference. And certainly with the pharmacy students going out into the world and being kind of healthcare professionals and understanding and being able to demonstrate that knowledge is something that kind of is very rewarding for me as an individual to have been kind of part of that kind of learning journey for those students.
Brilliant. Okay. So just kind of leading on from that question, what are you working on at the moment?
So in terms of research, so working on at the minute, teaching wise, so we're teaching pharmacokinetics at the minute. In terms of research, as I mentioned, I'm a neuroscientist. So in the lab, we're looking at Alzheimer's disease. So we're interested in how the cells in the brain change form and function as a consequence of the disease. And our hopes are that this will offer much better interventions for Alzheimer's disease. So at the minute, it's pretty much a blank canvas. There's been a lot in the news about some new drugs which haven't been approved yet in the UK, but that could change the landscape. But it's thinking about, right, what's next in terms of those new drugs and new medicines. So we're in the laboratory looking at different molecules and how they can change the course of that disease in terms of dementia.
Okay, brilliant. Right. Well, thank you ever so much for that, Mark, and thank you for everybody who's put in questions in the chat, and to everybody watching in school. I am just going to pass over to my colleague Lisa now, who's just going to finish off.
Thank you, Sophie, and thanks, Mark. I'm sure the audience has been inspired by your talk and understands more about why this common medicine. And also, I'm sure that most of us, as you said earlier, do have it in our medicine cabinet somewhere. I certainly have it in my handbag all the time. So, yeah, it's been really, really interesting. Please take a look at our website, thestemhub.org.uk. We have a huge range of pre-recorded content created by STEM ambassadors like Mark from various industries that you can watch on demand. So thank you for joining us today. Enjoy the rest of British Science Week, and hopefully we'll see you again in the future. Goodbye.